Hello everyone and welcome to another lecture of Physiology for Engineers with Professor Gonzalez-Fernandez at Lehigh University. Today we are going to focus on muscle physiology, and we are going to talk about the skeletal muscle, which is the muscle that is going to help us walk, run, carry weight, and we can action it in a voluntary way. I wanted to remind a bit the anatomy of the skeletal muscle. So we have different subdivisions. So here we have the skeletal muscle, for example, over biceps or triceps. This skeletal muscle is composed by muscle fascicles, and then the muscle fascicles, they are composed by muscle fibers. And inside the muscle fibers, we have the most important subunit, which are the myofibrils, okay? So the myofibrils are really important because they have different components and they are going to be involved in the process of muscle contraction. The first component that is really important here in the myofibrils are the thick and thin filaments. The thick filaments are the myosin, and the thin filaments that are the actin. Then the second component that we have to have into account for understanding the muscle contraction process is the sarcoplasmic reticulum. Okay. And finally, the last component of the myofibril that I wanted to go through is here, the T-tubules. TT or T-tubules. Okay. Okay, all of these components here, they are going to be very important for the activation of our muscle and muscle contraction, right? But in order to contract our muscles in a voluntary manner, we also need a neuron which is going to transmit an action potential and then it's going to activate the muscle, right? So we need a neuron here. Okay, so these neurons are going to be motor neurons. Specifically alpha motor neurons, and these neurons, they are efferent neurons because they are going to transmit the message from the central nervous system into the rest of our body. Okay, and these motor neurons here, the neurotransmitter that they secrete is acetylcholine, and therefore the receptors here on the muscle fiber, they are going to be nicotinic N1 receptors. And the part or the complex of the binding of the neuron to the muscle fiber is called the neuro-muscular junction. Okay, so we are going to look at this process a bit more in depth and everything that happens here in a neuromuscular junction. And this way we are going to understand the excitation-contraction coupling, okay? So let's get this a bit bigger here. So we are going to have our motor neuron with the acetylcholine. And then we are going to have our myofibril membrane here with our receptors, the N1 receptors, nicotinic receptors. And then we are going to have these T-top tubules. And then between the T-tubules, we are going to have the other component that we have seen here, which is the sarcoplasmic reticulum. And finally, we are going to have the thin and the thick filaments, the actin and the myosin filaments that are going to form the sarcomere. Okay, so here in red we have the thin filaments or actin, and then in purple we have the thick filaments or myosin. And all these two filaments, they form the sarcomere, and here we have also the M band, okay, which is the center of the sarcomere. Okay, so let's look a bit at what happens here and how the excitation contraction coupling works. So first of all, an action potential comes here, and what is going to happen is that the motor neuron is going to release here the acetylcholine, and then the acetylcholine here is going to bind to the N1 nicotinic receptors in the membrane of the myofibril. Okay, so this binding of the neurotransmitter to its receptor is going to activate here the sodium channel. So this is step number 2. So we have the activation of the sodium channel, and that's going to despolarize the membrane. So this depolarization is going to go into the T tubule. So remember these T tubules here, which are the third component that we talk about. And then this is really important because this depolarization is going to arrive to a very specific voltage-gated ion channel, which is called the DHP channel. So this will be a step 3. So the DHP channel is really important because it's going to be physically coupled to another very important channel here in the sarcomere. So when this DHP channel is activated, then this other channel in the sarcomere, which is a calcium channel, is also going to open. So this is 4th. What is going to happen is that all the calcium molecules in the sarcomere, they are going to leave through this voltage-gated ion channel into the cytoplasm of the myofibril. So then all these calcium molecules, they are going to come here to the sarcomere, and they are going to activate the cross-bridge cycle, which is going to activate the movement of the muscle and muscle contraction. Okay, so we have seen this excitation contraction coupling, again very quickly, acetylcholine release, then opening of the ion channel, of the sodium channel, then the polarization of the membrane, then activation of the DHP channel. We also open this calcium channel in the sarcoplasmic reticulum. The calcium is released into the cytoplasm, and then the calcium is going to initiate the cross-bridge cycle. So before we go into the cross-bridge cycle a bit deeper, I want to go through a couple of concepts here. So let's look at the excitation contraction coupling, right? So what we talk about is that we have the action potential and the release of the acetylcholine, and then the depolarization of the muscle cell membrane. So this is going to produce the depolarization, so more positive charges in the cell membrane. Okay, do this plus 30 millivolts. But then the cross-bridge cycle, so the contraction is not immediate. So let's say that this happens at time 0 milliseconds. So this will be time. So if we also have another graph for contraction, and here we have tension, we have that the muscle contraction doesn't happen immediately after the depolarization of the cell membrane. That happens a bit later, at time 40 milliseconds. Okay? Okay, so now we have done with this part. Let's move a bit deeper into the cross-bridge cycle that happens here in sarcomere. I'll see you in a second.